and I'll be your host for this session. And as we all know about the wastewater and environmental surveillance, it has been done globally to monitor and analyze the wastewater. And it is crucial in many ways that we have to identify, it help us to identify the potential health hazards. It help us to monitor the effectiveness of the wastewater treatment plants. And it can help us to generate some evidence, which can be further used to for policy making and some for some interventions. So in this webinar, we will explore two main areas. What is the significance of the wastewater and environmental surveillance, and what are the different global models? And this will be followed by some discussions on the role of the genomics in the wastewater and environmental surveillance. So for this particular webinar, we have a group of esteemed speakers with us. And thank you for joining us today. And we look forward for exploring this critical topic together with all of you. And first, I would like to introduce our first speaker for today, Dr. Paul Pronyk. He is a professor of Global Health Director, Duke National University of Singapore from Center for Outbreak Preparedness at Singapore. Thank you, Dr. Paul, for joining us today. And he is going to speak about the significance of the wastewater and environmental surveillance and some of the examples from the global models. So over to you, Dr. Paul.
Thanks, Dr. Pronyk. Good to see you again, as well as other colleagues. I'll just be sharing. You get it, Vincent? Okay, we're doing a tag team between myself and Vincent Pang, who's an epidemiologist working with us on wastewater-related work. So just to kind of kick off initially, we are with the Center for Outbreak Preparedness at Duke-NUS Medical School, and we act as a secretariat for something called the Asia Pathogen Genomics Initiative, which brings together countries across the region to really accelerate the use of genomics for a number of different use cases. And one of the big ones really is wastewater. And we draw together expertise across about 14 countries in the region to try and accelerate learning in that space. And I'll be presenting some of that work to you here today. Next slide. So, as we all know, we are in a bit of a hot zone, right, in terms of outbreak risk. Not surprisingly, issues of health system inequalities, dense populations, high levels of mobility, human-animal interaction, climate risk, all of these things really come together to really accelerate risk in the Asian region, and early detection is absolutely key. Next slide. So within the context of Asia PGI, the way in which we kind of think about how we as kind of almost a meta institution can support issues around early detection and surveillance, there's basically 4 different work streams that have emerged. The first is around kind of capacity development for early detection. And we focus on genomics and bioinformatics, but obviously that needs to be embedded within strong surveillance system with the strategic use of conventional diagnostics as screening tools usually. We support health system enhancement, which means planning and budgeting for surveillance and laboratory systems, and trying to ensure some of these new technologies can be accessible to folks in low and middle income country context, because prices are really coming down quite a bit. Assays are getting more sensitive across a range of different pathogens. So instead of doing a different test for different things, you've got basically one test that can address a range of different challenges simultaneously. We also look at enabling factors, which are kind of legal, ethical, regulatory issues around things like data privacy, data sharing, the balance between open access to data and sharing the results of new products that are developed from that, which is a big part of the pandemic treaty negotiation. So we have a legal team that works on that. And then finally, it's how do you take a lot of new insights that you glean from genomics and have it inform the development of new diagnostics, therapeutics, and vaccines. So the approach that we're using kind of takes something that even 5 years ago was often a research tool in many countries to look at rare diseases. And how do you apply that to high priority pathogens that exist in Asia and Pakistan, right? There's a range of different challenges that we all face. And you only want to do things like genomics on usually a portion of them. So that's quite key, finding what are the priority pathogens in your context. Secondly, looking at a One Health approach that looks at humans, the human-animal interface, as well as the environment, which includes wastewater and also airborne surveillance. The development of cost-effective delivery systems, so trying to put the right equipment in the right settings, whether it's centralized or decentralized, urban, rural, and so forth. And then obviously the goal of this all is developing real-time data that can inform a coordinated public health response. Next slide. So this is just a quick list of some of our partners. So in the space in which we're working, it really brings together, even in Pakistan, an interesting combination of researchers who often have been doing this work for a long time, particularly around genomics, and national public health institutions who really have a mandate for surveillance. And unless these groups kind of come together, it's going to be hard to really move quickly in what's becoming quite an important area. And this just shows the list of folks that are involved in that in the areas that we're working in. And again, it's mostly academic and public sector partners, but in some settings, the private sector plays a big role, and in other settings, NGOs might play an important role as well. Next slide. So part of the work that we're doing, which is something that we're doing in partnership with our colleagues on the call, with Dr. Afrinish and Dr. Imran, is establishing a regional expert wastewater working group that supports knowledge exchange in the region, the development of, taking global guidelines and saying, what does that mean in the Asian context? And trying to help us define what are our priorities as a region that we can work collectively towards. And that includes dedicated training in wastewater-related surveillance, including genomics and bioinformatics as relevant. Trying to look at what are the optimal strategies to adopt in different countries based on the settings and the priorities. We strongly support wastewater innovations, and that's things like different types of filtration and collection techniques, such as nanobeads, and different types of assays that can look for many pathogens at the same time, such as targeted next-generation sequencing. And then we provide direct support to countries on the ground, and that includes Pakistan, Cambodia, and Indonesia. And a lot of this work is supported with grants from the Bill and Melinda Gates Foundation. I guess now it's called just the Gates Foundation recently, as of January. Next slide. So this is just to say wastewater surveillance has been around for a very long time. It's one of the kind of historical tools that we use to look at polio and still using, obviously in many contexts, but it's kind of just an old paper from 1940 that highlights when it was first detected and how it's been useful. Next slide. As we know, we're on the cusp of polio elimination, although I hope that that's not set back too badly given some of the withdrawals in funding and so forth. But we know that there's currently sort of 500 environmental sampling sites across some of the high-risk countries. And so there's already systems in place to kind of collect this work and do it. So how do we adopt a multi-pathogen approach to have a broader canvas or a broader set of things that we're looking for in the context of wastewater? Next slide. From COVID, we've learned, of course, and I think probably folks know this, that it's an important early detection tool. By sampling wastewater, we get about a week or two at least head start before we start seeing clinical cases. And we saw this for COVID, but you see this for a whole range of things. In South Africa recently, they were showing some data that shows you can, even something as transmissible as measles, where you think you would pick up clinical cases very early, you're seeing it in the wastewater sometimes long before clinical cases appear. And I guess that's one of the important points here is that in areas where there are not robust clinical detection and surveillance programs, wastewater is even more important in terms of picking things up early. Next slide. And I think what this slide just tries to get at is that all of our sort of wastewater surveillances and One Health surveillance needs to be embedded within a multi-source surveillance system that draws together not just what's happening at wastewater, but obviously the primary care level all the way through to our tertiary care systems, depending on what you're looking at. In addition to that, the need to kind of engage with our animal health laboratories to try and understand, what are some of the risk environments that we're working at there. Next slide. Again, very similar sort of point from this slide that, when we look at, all infected people, wastewater is really one of the first places where infections will appear. And it's not just, it can be from feces, it can be from urine, it can be from sputum when people cough, and people actually, you can sample tuberculosis in wastewater, believe it or not. So there's a whole range of things that you can get from those samples that really do inform things that happen farther up in the pyramid before cases and hospitalizations and deaths become evident. Next. So we have spent some time looking at what are the priority pathogens in wastewater. And believe it or not, this looks like a big list, but it's not that big a list. So it's really only about 30, 35 or so pathogens that are feasible to detect in wastewater, right? Technically, that are actually actionable. And the biggest list of, the thing with wastewater is especially with metagenomics, you can detect all sorts of things. And it can be very confusing to interpret the results. So what's important from this slide is just to really hone our focus in onto things where if you get a hit, there's a very clear action that can follow from it, whether it's vaccine preventable disease, issues around being able to use medicines, dengue, so related vector control strategies, as well as non-pharmacological interventions such as isolation or identification of high-risk settings, as well as a special emphasis on identifying pathogens that might have pandemic potential. Next slide. Good. And I guess one of the purposes of this talk, which Vincent's going to take over in a bit, is really looking at how do you tailor and adopt a wastewater surveillance system? Where should it bolt in your particular context? So one of the first questions you've got to really ask is, what are the questions that you're trying to answer with wastewater? Are you trying to detect a new pathogen, so something very rare, like polio, for example, or an emerging virus of pandemic potential? Or are we trying to track variants? So wastewater is often used to look at, say, antimicrobial resistance and things like, cholera, typhoid, and so forth. Or are we using it to monitor intervention effects? And that's always a very interesting one because you can, again, as I was saying with the measles example in South Africa, you can see things in the wastewater, with measles, you often have some estimate of how you're doing with coverage at the population level. But as we know, in many of our contexts, we may not have a good denominator. So if we immunize 1,000 children, we still don't really know for sure what the coverage is, right? And there's very little information between that and cases being evident in our primary care clinics or in our hospitals. So wastewater provides like a quick snapshot of whether we're doing okay in terms of creating herd immunity and whether there's ongoing transmission happening in populations, which can then help us tailor and target, say, additional top-up immunization campaigns, or also see whether or not an immunization is working. If you've introduced a new immunization, say like pneumococcal conjugate vaccine, wastewater can give you insights into things like immune escape. The next step, of course, is what action do you take? And that speaks similarly to the slide I presented earlier, is that, you really want to, when we work with countries around which pathogens to prioritize, the first question we ask is, what do you want to do with it? If you find something, what does it mean? The next thing is really around issues of understanding what, when you're gathering a sample from wastewater, what population does it affect, or does it capture? And that is complex in different contexts. Sometimes you're gathering wastewater samples from a river. Sometimes you can gather it from a single institution. So you need to be really clear about what area you're sampling and what it represents in your particular context. In addition to that, in interpreting the results from wastewater, you need to understand issues such as whether there's animal contamination of water, whether there's spillage or seepage, where the samples get diluted by rainwater or industrial waste, and then how you use that information to interpret the results. Next question is really around, is it even feasible to detect the pathogens? And that is the slide I showed earlier. We've tried our best to kind of draw together the literature in terms of that longer list of, what are the things that are technically feasible to detect. And then based on these factors, as you're thinking through with your epidemiologists and with your surveillance programs around where to position wastewater, it's how do you do that in kind of the most cost-effective and sustainable way? Next slide. Great. Over to you, Vincent, on this one.
Thanks very much, Paul. Very good morning, good afternoon to everyone. My name is Vincent. Thanks very much, Dr. Afinish and Run for organizing this webinar to showcase the importance and the significance of wastewater surveillance. And I would just like to highlight in these slides the conceptual diagrams of how wastewater surveillance typically look like. Wastewater surveillance is a system that collects wastewater, mainly primarily from wastewater collected at the sewage pipes, which can be closed, semi-closed, or open systems from either the highly densely populated areas or the sparsely populated areas. And it could be also collected from the wastewater treatment plant as well. So following the samples collections, with the transportation of the samples properly to the laboratories, samples will be actually concentrated, the wastewater will be concentrated to allow higher chance of detecting some of these pathogens of interest, depending on the priority of pathogens of interest in the respective countries or in the respective provincial or national level. And following that, you have a concentration methods that allows the samples to be collected either through PET or some of the centrifugation methods. With the extractions of the genetic materials of these pathogens, with as much as possible the removal of the human genome genetic materials. And following which you have the detection using RT-qPCR or RT-digital PCR for early detection using primers of well-validated sequences to detect pathogen of interest. Nevertheless, there will be some form of challenges along the way. Some of these challenges, as you can see from the slides, in terms of false negative, you may not actually be able to understand why actually some of these results are false negative, and it could be due to poor rep sampling of the samples. The timing of the sampling process is not done ideally, mainly because most of the residents may not be available or around in the household, and therefore you are collecting samples that may not contain any form of fecal materials that have pathogens of interest. Generally, the timing of the sampling is within the morning time and the evening times when most of the residents are back home. And in terms of false positive, there will be some potential sample contaminations from the way how the other industrial discharges or environmental contaminants may actually affect the integrity of the pathogens that you are actually testing on. And in terms of concentrations, there may be a need for trial and error on which methods would be more efficient in terms of concentrating some of these water samples. And in terms of false net positive, there could be cross contaminations if we are actually collecting more than one samples from different sources. So extraction wise, there's a need to have a bit of trial and errors on what methods will allow you to do extraction efficiently of the genetic materials of the pathogens. So there are a couple of number of different kits available that for different pathogens that you may need to try out because different water conditions may affect the buffers of the quality of how the extractions can be done of the genetic materials. Likewise, the false positives can actually be affected by the reagent contaminations. And lastly, in terms of the detection tools that we use for RT-qPCR, there's also a need to invest some time in understanding which methods would be actually most appropriate for the usage of the water samples that you're collecting from. And some of these the PCR may not be as sensitive to the low viral load of bacteria, viral load that you're actually collecting from the wastewater samples. So likewise, for false positive, you have non-specific assays. So there are some other key considerations for sampling designs. There's recommendations from WHO. If resources allow, so we should actually collect more frequent sampling so that we have more rapid turnaround of results, providing better early warning signals. If possible, we should actually close down to final spatial span sampling scales so that we are able to identify more targeted at-risk populations for more targeted public health actions and to be promptly executed to minimize the risk of transmissions. We are also, WHO is also recommending safeguarding high-risk settings. So samplings done at aged care facilities, dormitories, prisons, military sites, border controls sites, and sites with animal and human environment interface are highly crucial for samplings with wastewater surveillance, as the signals may actually provide us very prompt understanding of how transmission may occur, and public health interventions may actually promptly start to indicate some of these transmissions. So further recommendations from the European Union, HERA, Health Emergency Preparedness and Response Authority, they recommended at least wastewater from large cities with about over 150,000 inhabitants to ensure efficient use of the resources for peacetime monitoring of priority pathogens of sampling frequency 2 samples per week. And additional sampling sites can be actually increased depending on the epidemiological understanding of the disease of interest based on the at-risk profiles of the populations as well as the at-risk activities of the populations, which includes the vulnerable communities. And samples should be taken at the inlet of wastewater treatment plants or upstream at the wastewater collecting network, minimally as a baseline, to have a very good overview of what are the potential infectious disease transmission that may be occurring at the residential areas, especially when vaccination coverage are actually not high enough to deter any form of outbreak of occurrence. And if possible, for early warning surveillance purposes, the results of the samples should be recorded and reported to the respective national authorities as well as possible, not later than 48 hours. So we are all very concerned about the next potential zoonotic pathogens, and therefore there's a high recommendations of sampling at the human environment zoonotic interface, animal interface, for some of these potential spillover from animal to human populations. And there has also been recommendation to adopt the use of next-gen sequencing, either in the form of targeted sequencing approach or a metagenomics approach. So WHO has also recently produced a guidance draft on implementation and prioritization and integration of wastewater surveillance in member states. They have considered some of the key factors for implementation and prioritizations, looking at the target pathogens, what would be the prioritized pathogens of interest that national authorities should consider, as well as the context of leveraging on the use of wastewater surveillance, such as the epidemiological situations of certain diseases of interest, as well as the capability of testing in terms of technology, reagent accessibility and availability, as well as the reporting mechanisms. And they have also recommended the use case of documentations for future refining of the mechanisms for wastewater surveillance implementation. And in terms of wastewater surveillance program element, they have recommended either it could be done on a routine basis or a more targeted time-limited basis, which is known as a agile surveillance approach, depending on the resources available. So this is a regional dashboard mapping of wastewater surveillance initiative in Asia, where Asia Pathogen Genomic Initiative is very interested in understanding and what are the activities that has been in terms of wastewater, using wastewater to sample priority pathogens in Asia. So this is a QR code that you can actually scan and provide your inputs in terms of wastewater surveillance activities and initiatives that you have actually created in Pakistan, for our understanding and for better learning and sharing of knowledge in the near future. So just to share briefly what have been collected from the data that this dashboard mapping have provided in terms of helping us to understand the characteristics of the wastewater surveillance activities and initiatives in Asia. So far, we have about 45 institutions in Asia, in 19 countries, of about 89 projects. And among these projects, it is noted that a majority of this wastewater surveillance has been very much focused on one pathogen, detection of one pathogens. And in order for efficient use of resources and the advances in technology, as well as the reduced cost of these advances in technology, such as next-gen sequencing, as Paul mentioned, there's a great need and understanding that we should actually adopt a multi-pathogen approach in implementing wastewater surveillance. So these are some of the sampling design characteristics in terms of sampling frequency. Mainly, most of the projects are actually conducted monthly sampling and weekly sampling. And in terms of sampling locations, they are mainly still from the wastewater treatment plants, covering very broad intelligence of the potential transmission of wastewater priority pathogens in broader residential areas. Likewise, in some of the rivers, open lakes, water bodies, and streams, and unsewage discharge ponds. So, and in terms of priority sites that we have noted, mainly are covering residential sites and high-risk institutional settings. So some of the priorities for implementing wastewater surveillance, which we have recognized and discussed a lot intensively, especially during our last year wastewater surveillance symposium in Asia, co-organized by Gates Foundation, as well Bill and Melinda Gates Foundation, as well as Prepare from our Ministry of Health in Singapore, as well as Duke-NUS Medical School with the Asia Pacific Genomic Initiative, and as well as National Environmental Agency. We noted the need for more funding to sustain a lot of the good work that has been implemented during the SARS-CoV-2 pandemic, leveraging on wastewater surveillance as a tool for early detection. But at the same time, we have responses from the participants looking at the need for more governmental support in sustaining these good practices of implementing wastewater surveillance for the country to detect, as an early detection tool for priority pathogens, especially the vaccine preventable ones, where vaccines availability can be used to reduce the risk of transmissions. So there are also some of these characteristics in terms of utilities of wastewater surveillance that have been captured in the response that has been submitted through our dashboard. They have been mainly used for early disease detection as well as identification of high-risk settings. And interestingly, we really hope to reduce, the incidence of, not leveraging on the wastewater surveillance data to make any form of public health decision making. And I think this is an area that we are seriously working very hard to engage stakeholders from different countries in the low and middle countries, where wastewater surveillance are available for them to actually inform public health decision making. And we are ready to actually prepare them in terms of in the form of training, as well as sharing of educational materials, protocols in the laboratories, and through some of the webinars that we are also organizing with some of the partners in Asia to better inform how we should leverage on the wastewater surveillance data for public health decision making. So just summarize, in terms of regional advocacies, we are recognizing that the One Health and multi-pathogen approach in implementing wastewater surveillance remains very limited in Asia, but it's really crucial for us to leverage on One Health and multi-pathogen approach for early detection of potential spillovers of zoonotic pathogens. And we really need, and we are actually very actively working towards creating this regional coordination effort in Asia Pathogen Genomic Initiative, as well as trying to compile a harmonized protocol for Asia that can be used as a reference that needs to be validated in respective countries, nevertheless. And it is actually important for countries to implement and sustain and expand wastewater surveillance beyond SARS-CoV-2. And we really encourage and will definitely leverage on the resources and the network that we have in Asia Pacific Genomic Initiative to support translation of wastewater surveillance data in terms of interpretations of data, as well as identification of a threshold for public health actions remains limited. And we definitely, it's important for us to close some of these gaps with the national stakeholders to maximize the investment and the sustainability of wastewater surveillance in the country. So with this, I would like to end off by highlighting that in Asia, 60% of the world population remains vulnerable because we have the higher risk of outbreaks globally, one of the highest region in the whole world. And Asia PGI, as Paul has highlighted the role, we leverage on cross-country collaborations, and we would love to engage many more national stakeholders in terms of capacity development and education, and hopefully we can sustain wastewater surveillance for early pathogen detections in the near future to come. Thanks very much, everyone.
Thank you. Thank you, Dr. Paul and Vincent, for a very informative presentation. And we really like the ideas, and we totally agree with you on the number of the aspects that have been discussed in this presentation. And we thank you for being with us. And we look forward to the question answer session by the end of this webinar. And I think we can go towards our next presentation for today. We have Dr. Rehan Zafar Paracha with us. He is a senior scientist, and he's been a huge experience in the field of genomics and bioinformatics. And he is working with us at National Institute of Health. And he is going to discuss about the role of the genomics in the wastewater and environmental surveillance. So over to Dr. Rehan.
Thank you very much for giving me the time. Thank you very much, Dr. Paul and Dr. Pang, for a very nice presentation and very nice discussion about the requirement of the day and the insightful discussion about the requirement of wastewater analysis and analytical workflows which are required and the efforts of the Asia PGI. So taking the presentation a bit forward with the role of genomics in wastewater environmental surveillance, that what is the important area of consideration in analyzing the wastewater, and why we are considering that area of role of genomics. Suppose we can test a lot of other things as well, but the genomics role is important to understand that what's going on. Okay, so again, Dr. Paul, Dr. Afrinish, Dr. Pang, Dr. Judith, and Dr. Imran, thank you very much for being here and for making such a platform in which we can analyze and understand the surveillance of different bacteria and their importance in AMR and different other perspectives. So what is genomic analysis in wastewater? Like how we can define it? It is the genetic material analysis like DNA, RNA of microbial communities which are present in wastewater. Why we are interested in the microbial communities? Why we are focusing towards that? Because characterizing the microorganisms and their functions is the potential and the focusable area which can lead us towards saving a lot of lives before the actual pandemics or endemics. How we can do that? The simple answer is by using the next generation sequencing, RT-PCR, digital PCR, metagenomics, and whole genome sequencing. What is the goal? So the goal is to understand the microbial diversity. Like we can understand the functional capabilities. If we know about the particularities of the bacteria, we can know about the functions of the bacteria, and with the sequences of the bacteria and microbial environments in hand, we can test that whether something bad is going to be happen or something pandemic is going to be happen or not. In this sense, antibiotic resistant genes is the most important thing which we are looking for in the sequences and in the genomics of the microbial diversity of wastewaters. So this is the pictorial representation, so that how we are going to fetch that DNA sequencing or the RNA sequencing out of the microbial diversity of the wastewater. We will get the DNA and we will extract it. Then after that extraction, we are moving towards mostly towards the shotgun or amplicon sequencing. In this amplicon or shotgun sequencing, we are ultimately identifying the common genes within that community and what are the differences and how these are going to affect the protein later on. from the genomic sequences. Because the sequences of the microbial communities in the wastewater are now with us, now we can develop that what's going on in the Pakistan and different areas which are being under consideration by the Asia PGI, and how we are going to deal with this. So wastewater is a reservoir for pathogens, as have already been told by Dr. Paul and Dr. Pang, in which a lot of bacteria, viruses, and parasites are present. So the public health monitoring is one of the first focus area, that why we are going to analyze the microbes and how we are dealing with the genomics. Like I want to show it from this data tracker, like which are present by the which are offered by the CDC. By looking at this, we can understand that what's going on with the genomics level analysis, and how the things are being covered at different levels. Here you can see the wastewater analysis and the COVID-19 national trends, which are being reported by the CDC and on their dashboard. So whenever you have the sequences in hand out of the wastewater, then you can make such kind of analysis, like what are the trends in positive test positivity, what are the trends in emergency department in relation to the presence of certain bacteria and certain pathogen in the wastewater. What are the hospitalization rate and trend in percentage COVID-19 deaths? All of these things are then relatable to each other. So there are some kind of problems over here in the loading of the page. You can see this link at your side, that what are the things which are being happening in this kind of analysis whenever you have the genomics in. In the same sense, antimicrobial resistance is another thing which for which we are going to analyze the overall genomics in our hands or in the wastewater. As these links are also from the CDC site, may contain some kind of a problem. So I have included one of the graphs, and luckily that kind of graph is also presented by Dr. Paul in his slides. Here you can see that the viral copies in the against the time and the titer of the copies are being recorded by the universities and the CDC over the period of time from September 2024 to February 2025. What are the patterns? You can see here the patterns, that the wastewater, whenever the virus copies are present in the wastewater, then the same hospitalization rates are going to be correlated with it. Like there is a peak of viral copies in the wastewater, then there is a peak of hospitalization as well. And you can see the ICU admissions at that time as well. So you can correlate between the genomic copies and the hospitalization rate or ICU admissions, and you can form up a lot of things in relation to the like a lot of policies in relation to the presence of certain copies, certain variants of the bacteria and the pathogens in the wastewater. This is also taken from one of the paper in which they are monitoring the global presence of antimicrobial resistance genes in the metagenomics analysis of urban sewage water. They have considered some of the areas like Africa, Asia to South America, and they found that the antimicrobial resistance is mostly present in the Africa, in the South America, and now the Asia is going to be up in this kind of a problem. So urban sewage water, antimicrobial resistance, and the presence of the antimicrobial resistance genes. Definitely, we are getting interested in such kind of a sense that what type of antimicrobial class, resistance class is present in all of these samples. So here are some of the AMR classes against which the lot of bacteria are being presented over here from different samples which are resistant against these AMR classes or the antibiotics. Likewise, we are also interested in observing that kind of effect, that what are the AMR genes which are affecting overall the population and are present in the wastewater as well. And these genes and the classes could not be identified until and unless we are doing the genomic analysis of our wastewater. So environmental safety early warning systems are some of the things which have already been discussed by Dr. Paul and Dr. Peng in their slides. And these are being possible only with the help of the genomics and their analysis in the sewage water and the wastewaters. How we can particularly are going to do it by using the approaches of metagenomics in wastewater. So metagenomics is a technique that sequences all genetic material in a sample, providing a comprehensive view of microbial communities. What are the advantages? Because we haven't to deal with particular things, and we can scan all the genomes which are present in the wastewater. So it will make us aware. It provides a platform basically so that we can scan all the genes. We can detect pathogens, AMR genes, microbial functions, and track the spread of infectious diseases. There are a lot of other advantages as well over the traditional methods, which can be discussed later in the next few slides. So particularly, we are interested to go with both of the ways which are being offered by the metagenomics, like shotgun sequencing and amplicon sequencing. In the amplicon sequencing, you can see over here that particular areas of interest are being sequenced again and again to analyze that what are being happened, what is happening in the particular area of interest in the genome. Whereas shotgun sequencing relates with all the sequence of the bacteria and all the bacteria which are present in the wastewater. So we are going with the shotgun as well as the amplicon sequencing of the wastewater. Out of that metagenomic analysis, we are interested in alpha diversity, beta diversity. What is the taxonomy of the group? What are the OTUs and functional units which are being present in the wastewater? How they are making the networks? How they are? What are the phylogenetic analysis of those bacteria and those classes? How we can classify them in the AMR or non-AMR groups? What are the taxonomy difference comparisons? So all of these things would be understood once we will get the genomics and once we will get through the metagenomics of the analysis. So metagenomics is detecting the mobile genetic elements like ARGs. We can detect that as well. We are interested in monitoring the hotspots for resistance development, such as there are some areas in wastewater treatment plants where diverse microbial communities interact with each other. And with the metagenomics, we can provide data for global surveillance systems to mitigate risks associated with resistant pathogens. So in the summary, genomic analysis of wastewater is a powerful tool for monitoring public health, understanding environmental impacts, and understanding antimicrobial resistant genes, which is the requirement of the day. And how we are going with it. There are different tools, and metagenomics is one of them. Thank you very much. These are all the slides from my side.
Thank you so much, Dr. Rehan. It was really interesting to listen from you and learn from your experiences in the, particularly for the metagenomics and the genomics. And we will definitely seek your expertise in the project as well. And as we are running short of time, I think we can quickly go towards the panel discussion. We have 3 panelists with us. And first, Dr. Vincent. He is the assistant professor from Duke National University of Singapore. Then we have Dr. Judith Wong. She is from Microbiology and Molecular Epidemiology Division, National Environmental Agency in Singapore. And then we have Dr. Imran Nisar. He is Associate Professor, Vice Chair, Department of Pediatrics and Child Health at Aga Khan University in Karachi. Thank you for all of you for joining us in this panel discussion. And we have some points for you. And first, I would like to go with Dr. Judith. I hope you'll be doing fine. Thank you for joining us. So given the immense potential of the wastewater surveillance to monitor various infectious diseases, what are some considerations in implementing a wastewater surveillance program for public health?
Right. Hello. Hi, everyone. And thank you very much for the chance to share our experience. So with regards to Afrinicia's question.
Dr. Judith, we
Yeah.
cannot hear you.
Oh, you can't hear me. Hang on. Are you able to hear me now? Can you hear me?
Yes,
Hello?
you are audible. Yes, you are.
Okay, great. Okay, thank you. Yeah. Okay, thanks very much, Afrinish, and thank you everyone for the chance to share our experience. So with regards to the question, I believe what Paul and Vincent presented earlier on gave very good consideration points. The first is really understanding the sewage network that we are looking at, and the population coverage that we can monitor. So that will then frame the pathogens that we want to monitor. Of course, the feasibility is a consideration. I think one question that I would want to encourage anyone starting a wastewater program is the so what, right? Like, why are we doing the testing? Because in principle, you can actually test many different infectious diseases, and you would be able to get a trend of the transmission, for example. But if we go back to the purpose of the surveillance, if there is an existing clinical surveillance which is already very good, for example, if SARS-CoV-2 monitoring is already being done in the clinical settings, or monitoring some diarrheal diseases is already being done, then the question is whether or not the wastewater surveillance adds any value. If it does, then by all means do it, especially if it's complementary or it provides more information about transmission trends. But if it does not, it doesn't mean that we need to test every pathogen just because we can. I would like to highlight one, maybe 2 examples in Singapore, which we have considered. So for example, the monitoring of respiratory pathogens. While we are able to do this in using the wastewater matrix, we are aware that in Singapore, we do have a rather decent clinical testing regime. So we actually do not do this on a routine basis, given that the clinical surveillance is sufficient. But our program is set up such that we are able to step up should we see any interesting threats or important trends coming about. So that's one example for respiratory viruses. But another example is that of Zika testing, for example. Because we know that the clinical manifestations are generally mild, clinical surveillance may not pick up all the diseases or all the cases. So wastewater testing then provides a non-intrusive approach to monitor the trend of transmission and to monitor if there are cases. And this is especially important for Zika because although it causes mild disease, it actually causes severe outcomes in a subset of the population, such as pregnant women and their unborn fetuses. So these are just some examples of how we would consider implementing a wastewater surveillance program. It's really important to consider the population, what is already being done for these different infectious diseases, and then balance the surveillance approach. Thank you.
Thank you, Dr. Judith. It was really interesting, and we totally agree with you that how we can go towards this implementing the program, and it seems a little challenge. And with this, I think I can go towards Vincent and ask the relevant question from him regarding that. We see that there's a huge importance of the wastewater and environmental surveillance programs. So, what would be the most important next step to institutionalize the wastewater surveillance systems? Like, how can we go into the institutions? Like,
Thanks very much, Dr. Afinish. And I think, Dr. Judith has raised up a very important point about, mapping out, extensively as much as possible the sewage systems. And I think beyond that, there's a number of other issues that and areas that may need to be focused on as well. Just want to highlight maybe two. First of all, it's actually in terms of at-risk populations. I think with understanding that in a resource-constrained setting, especially, there's a need to efficiently make use of the resources. And then I think one approach would be to identify vulnerable populations that may not have full accessibility to healthcare systems. As Professor Paul mentioned earlier, that wastewater surveillance system can be actually more valuable than clinical systems in the situation whereby healthcare services and accessibility and availability are actually really limited. And I think this is an area that we can make use of this opportunity to create a use case, in fact, an investment use case in the documentations to highlight the importance of wastewater surveillance in a scenario where healthcare systems are lacking, and also in a situation whereby vaccination coverage are not as ideal, due to be it geographical challenges or due to society, hesitancy for vaccinations. I think this is an area where wastewater surveillance can be the next step will be to identify some of these residential areas where it fulfills some of these conditions. And I think that will really fulfill and maximize the use of the limited wastewater surveillance resources that we have for a start as a next step. And then we can actually use that opportunity to engage the community in terms of trying to make sure that they are also well informed about the importance of wastewater surveillance and the need for wastewater collections from some of these residential areas. And we have also been looking at ethics as well as privacy issues, both for pathogen genomics as well as for wastewater surveillance. And I think it's an area that potentially may have been neglected during the pandemics, in the sense that because in emergency crisis situations, I think public health potentially outweighs the need for some of these considerations. But as we move into peacetime monitoring, I think there's increasingly a need and question, about the way how we collect wastewater. And I think we need the support from the community, especially the vulnerable ones, as highlighted earlier in situations where they make them more vulnerable, to maximize the use of wastewater surveillance. And hopefully, these are some of the next steps that we can, leverage on through this platform that with the stakeholders here. Hopefully, while also trying to increase our resources at the same time. So I think, one step at a time will help us to, push through the need for institutionalization of wastewater surveillance. I hope this helps. Thanks.
yeah, thank you so much, and I totally agree with you. This is the next step, or this is the best approach that a country or that a system can adapt to understand that it seems a little challenge, but this is the best way to address the system establishment. So with this, I go towards Dr. Imran Nisar. He has a huge experience of working in Pakistan and towards genomics and establishing various kind of systems. So talking about the challenges in the LMICs, like in country in Pakistan, resource limited settings, what do you suggest that how should we go towards, what are the basic challenges and how should we address those challenges to overcome, and how can we establish a wastewater surveillance system in LMICs?
Thank you, Afrinch. I think that's an excellent question. So, as it goes with any kind of surveillance systems, the wastewater surveillance systems can also be prohibitively expensive to establish. And I agree with what Dr. Judith Wong said earlier, that we have to always weigh in that if we have a good clinical surveillance systems, then the wastewater surveillance systems can act as a complementary system. It's not meant to be a replacement for the clinical surveillance systems, but it has its certain set of advantages as well. Having said that, Pakistan has a long established environmental surveillance systems for the polio, with a very wide network spread across the country. So that's also one way to think about it, how we can leverage upon that existing surveillance system to expand it to other pathogens beyond polio, including the wider list of respiratory and other pathogens. And one other thing to consider in the context of Pakistan is there's large areas in Pakistan which are still not served by a formal sewage system. So that presents its own unique challenges. Even when we have formal sewage system, those are the sewage networks are not clearly mapped, or those network maps are not accessible or present. And in the cases of sewage treatment plants, many a times those treatment plants are not functional. But many of these problems can be overcome by leveraging either on the existing resources, or building on sample surveillance sites on smaller areas. And the other thing I think, which is related to the amount of data that is produced as in terms of the bioinformatics output, and the limited resources and the capacity that might be present in many of these LMICs, and that holds true to an extent for Pakistan as well. But we are overcoming with because wastewater surveillance bioinformatics could be much more complex than the clinical bioinformatics because of the presence of multiple pathogens and everything. But there more and more so, there are like standardized automated pipelines that are being developed, which can be useful to standardize the analysis purpose as well. So I think in the end, we also need a strong financial and political support for this system to work. And I think we have a very good opportunity in the form of an existing environmental surveillance system for polio to leverage on that and expand it to other pathogens. Thank you.
Thank you so much, Dr. Imran. And I totally agree with you. The challenges are here, but with the combined approach and with the collaborative approach, definitely we can look for some solutions as well. And with this, I think we come to the end of our session. I really wanted to thank all my speakers and panelists for joining us for this session. And I can see a very good attendance as well for all the participants for joining us, listening to this critical important topic. If you have any questions, I think you can write in the chat box and my team is there. They will definitely respond to you, and the email address will be there. You can write to it that as well. And with this, thank you all for joining us today for this webinar. We appreciate your time and the engagement, and we look forward to connect with you in the future. Thank you so much.
Thank you very much,
Thank
everyone.
you.
Thank you.
Thank you, everyone.
Thank you. And a big thank you to the Duke team. Thank you.
ये ईमेल मैंने भेज दिया इसमें।